Good afternoon students, welcome to the online lecture series. So today I am going to take SYBAC paper number 1 of semester number 4, that is genetics. So today we are going to learn about the structure of RNA and DNA. It comes under the topic understanding DNA. So here we are going to start. Before starting my lecture, we are going to recap what we have learned earlier. So earlier we have learned about the various experimental proof which were given by various scientists about DNA as a genetic material. All we know that before concluding the DNA as a genetic material, various scientists put an assumption that it is a protein or it is a DNA. But many scientists prove, and later on DNA was proven as a genetic material of each and every cell. So we have seen the Hershey-Chase experiment which was done on the bacteriophage. We have seen its conclusion also, its experiment also. Later on we have seen that experiment on the plant virus, that is on the TMV virus. Experiment was carried out by the Gerer and Skarman, and they concluded that experiment in 1956. We have seen that experiment also and its conclusion also. The Franklin, Kernhunt and Singer, they also concluded an experiment which was on plant virus, and it was done after the experiment on TMV in 1957. We have seen its conclusion also. So our today's session, after learning about the various experiment, now it was proven that it is the DNA molecule which is the genetic material and which is going to pass from one progeny to the next generations. So today we are going to see the two types of nucleic acid, because when the TMV in the plant virus it was seen, it was studied that they lacked the DNA. In them the RNA was playing a vital role as a genetic material. So that was also proven. So both the nucleic acid in some or other cell were playing as a genetic material. So we are going to see the types of nucleic acid, that is the DNA and the RNA. And the structure of DNA means what the DNA is composed of, the nitrogen bases, the types of nitrogen bases, what are nucleosides, what are nucleotides, and the polynucleotide chain. Okay, so this is our session agenda for today's lecture. So about nucleic acid, we are going to start from the nucleic acid. We all know that there is the absence of the nuclear membrane in the eukaryotes, but prokaryotes have the nuclear membrane. And in that nuclear membrane, the nucleic acids, they are present. One it is DNA and one it is RNA. Only some viruses, they do not have the DNA. So in absence of DNA, there is the RNA which is the genetic material of that viruses. So nucleic acid, they are long chain polymeric molecules. The monomer, that is the repeating unit, is known as the nucleotide. A one nucleotide, it is bonded by a phosphodiester bond to other nucleotide, and a long polypeptide chain is formed. And that long polypeptide chain, it is supercoiled and it is incorporated in the cell in the form of DNA. So hence sometimes nucleic acids, they are referred to as polynucleotides because they are made up of many type of nucleotide. Nucleic acid, we know that they are the carriers of the genetic information. In all living organisms, the hereditary information is stored in deoxyribonucleic acid, that is DNA, which is the molecule formed from the repetition of the nucleotides making a DNA as a polymer. It is called as the deoxyribo because a absence of oxygen group at the third position of the sugar it is present. So it is called as the deoxyribo. Ribose sugar is present in both nucleic acid. And in RNA, the oxygen group is present. So here the name is called as the RNA, that is the ribonucleic acid, ribose nucleic acid. DNA, that is the deoxyribonucleic acid, because on the third position of the sugar molecule, the oxygen group is absent. So it is called as the deoxynucleic acid, which we are going to see in our later slides. The structure and properties of the nucleic acid. There are 4 different types of nucleic acid in DNA which forms a universal code for the hereditary information. Nucleic acid, that is RNA, the other kind of nucleic acid, a related molecule to a DNA, but it is single stranded mostly. It is also having the 4 type of nucleotide, but in RNA the thymine is absent, and in placement of thymine, a uracil is present in the RNA as a nitrogen base. It is also the polymer of 4 nucleotides, there of which the same in the DNA, with the 4 one is slightly different. It has many functions in the cell, notably acting as the intermediate between the DNA and the proteins. Some viruses even store their genome in the form of RNA molecule rather than DNA. Those who are having, example, plant virus, that is the TMV, it is not having a DNA. So, RNA is playing a genetic material in that cell. Structure and properties of nucleic acid. Nucleic acid, that is DNA and RNA, both are composed of the 3 molecules, that is a pentose sugar, a phosphate group and nitrogen bases. Sugar, it is deoxyribo in DNA and ribose in the nucleic RNA. So, it is on the name it is given, that is the RNA and DNA. Deoxyribose means the second carbon of sugar lacks the oxygen atom which is present in the ribose. Sugar in the nucleic acid, they are the backbone. Sugar and phosphate, both molecules, they are the backbone of the DNA molecule, and in between there are the nitrogen bases attached to each other with the hydrogen bonding in them. Chargaff rules, according to the Chargaff rules, the T is bonding with a A is bonding with a T and C is bonding with a G. Between the adenine and thymine, there are the double bonds present, and between the cytosine and the guanine, the triple bonds they are present. One they are purines and one there is pyrimidines. Purines are the adenine and guanine, and pyrimidines they are the thymine, uracil and the cytosine. So, in the nitrogen bases there are the hydrogen bonds present, and sugar and phosphate they are the backbone of the DNA which are linked together by the phosphodiester bond, which is a covalent type of bond, or which is also called a phosphodiester bridge. Now we are going to study about the DNA. DNA, it is the chemically DNA is composed of pentose sugar, phosphoric acid and some cyclic bases, that is the nitrogen. The sugar moiety present in the DNA molecule is beta D2-deoxyribose. The cyclic bases have the nitrogen in them, that is the adenine, guanine, cytosine and the thymine. These bases and their arrangement in the molecule of the DNA play a very important role in storage of information from one generation to the next generation. DNA has a double helical structure. We know that there is a central axis, and along the central axis the 2 helicals, that is from running from the 5' to 3' and anti-parallel strand, that is 3' to 5', they are coiled each other on that super helical or on this axis. Structure in which strands they are complementary to each other, means they are anti-parallel to each other. About the RNA, it is the RNA molecule also composed of phosphoric acid, a pentose sugar and the cyclic nitrogen bases. The RNA has beta D ribose in it as a sugar molecule. The heterocyclic bases are present in the RNA, that is the adenine, guanine, cytosine and uracil. Thymine is absent in RNA. Instead of thymine, it is uracil in the RNA. In RNA, the 4th base is different than of DNA. The RNA is generally a single strand which sometimes fold, and that results to a double helix structure. Now, RNA DNA is only of one type, but it has different forms. For example, A DNA, Z DNA, E DNA, D form of DNA. Z DNA, C DNA, Z DNA, it is the left-handed DNA molecule. But RNA have the 3 types. One, it is the mRNA, second one it is the rRNA, and third one it is the tRNA. mRNA, it is the messenger RNA, tRNA, it is the transfer RNA, and the rRNA, it is the ribosomal RNA, which plays a very important role in the further proceedings for the protein formation, that is the transcription and the translation. When a DNA is transcribed, a molecule of RNA is formed, which is the mRNA, which is a single-stranded molecule. And that single-stranded molecule, that is mRNA, it is translated in the form of long chain of amino acid by the tRNA and the rRNA. So, the various types of RNA play a very vital role in the formation of the proteins required by the cell for its various metabolic activities. Function of nucleic acid, both RNA and DNA. Nucleic acid is responsible for synthesis of protein which is required by the body. So RNA play a very vital component for protein synthesis. Loss of DNA content is linked to many type of diseases because it is the hereditary. It includes the hereditary information of your whole body or of the cell. DNA is essential component required for transferring genes from parents to offsprings. All the information of the cell is stored in the DNA, so it is very important part of your body, of the cell. DNA fingerprinting is the method used by the forensic expert to determine the paternity. It is also used for the identification of the criminals. So it is very important. Components of DNA, we have already seen that every nucleic acid, that is, it may be a RNA or it may be a DNA, is composed of the three main components, that is the nitrogen base, a sugar molecule and the phosphate group. So here we are going to see about the various three components of the nucleic acid. First one is the nitrogen base. It contains the purines and pyrimidines. Purines, they are double ring compound, and pyrimidines, they are single ring compound. Purines, they are the adenine and guanine, and pyrimidines, they are the thymine, uracil and the cytosine. They both are the organic compounds to take part in the synthesis of the DNA and also the RNA. Therefore, they are called as the building blocks of the genetic material. Now, you can see in the diagram that a purine, a parent compound, and the pyrimidine, a parent compound. Purine, they are double ring compound containing the various carbon, nitrogen, hydrogen components, and pyrimidine also, they also component, but here the amino group or the oxygen group has the distinct position. According to the position, the nitrogen base, it is going to have its name. So adenine, it is having the amino group at the 6th position. And guanine, the amino group, it has the 2nd position, and oxygen group, oxide group is at the 6th position. You can see the pyrimidine, they are the single ring compound which is having the various arrangement. In adenine, you can see the there are 6 number of carbons. So number of carbon, nitrogen and hydrogen and oxygen also varies on the nitrogen basis. What are the distinguished features of the purines and the pyrimidines? Purines, they are the heterocyclic aromatic organic compound composed of a pyrimidine ring fused with the imidazole ring. Pyrimidines, they are the heterocyclic. They are also the heterocyclic aromatic compounds which is composed of the carbon and hydrogen mostly. It comprises the adenine and guanine. It comprises cytosine, thymine and the uracil. It consists of 2 hydrogen carbon rings and 4 nitrogen atoms. It consists of 1 hydrogen carbon ring and 2 nitrogen atoms. The melting point of purine is 214 degrees Celsius and the melting point of pyrimidine is 20 to 22 degrees Celsius. The next component of nucleic acid is the sugar. So we know that the 2 types of sugar, they are present. One, it is the ribose, and second one, it is the deoxyribose. There are 2 types of sugar present in the nucleic acid. The sugar mobility present in the DNA molecules, that is the beta D 2-deoxyribose, whereas RNA has the beta D ribose mobility. You can see in the figure the carbon molecule in on the 2nd position of the deoxyribose, the oxygen group is absent. So it is deoxy and one, it is the ribose. Components, the 3rd component is the phosphoric acid. It is present in both, that is DNA and RNA, and the phosphate and the sugar molecule, they are attached each other by the phosphodiester bond and they are the backbone of your DNA. Phosphate group is attached to the 5th position of the sugar, pentose sugar. It is 5 carbon containing sugar, so it is called as the pentose sugar. You can see the phosphorus molecule having PO4, that is 4 molecules of oxygen. Now we are moving towards the nucleosides and the nucleotides, because we know that it is a polynucleotide chain. DNA, it is the polynucleotide chain. Poly means many nucleotides, they are bind together to form a DNA molecule, and they are bound together by the phosphodiester bond. And between the nitrogen bases, there are the hydrogen bonding. If these hydrogen bonds are broken by the various enzyme, example, in replication, the hydrogen bonds, they are broken down and the double-stranded DNA is converted to single-stranded DNA to do the replication. Here also, the nucleosides and nucleotides, they are bound together by various bonds. So first we are going to see about the nucleoside. The main difference between these two is the nucleoside, in the nucleoside there is the absence of the phosphorus molecule, and in the nucleotides the nucleoside plus the phosphorus molecule together have a nucleotide. A nucleoside, it is the compound that consists of a purine and the pyrimidine combined with the deoxyribose or the ribose sugar. A unit consisting of the nitrogenous base bonded to a pentose sugar is termed as a nucleoside. The first position carbon of sugar is linked to the nitrogen 9th position of the nitrogen base of a purine or a pyrimidine, and first position of the pyrimidine by the N-glycosidic bond. You can see a nitrogen base uracil and a ribose sugar, they are linked together by a glycosidic bond, and this is forming a nucleoside. In a nucleotide, there is the presence of the phosphorus molecule. A nucleotide, they are the building blocks of the nucleic acid. They are composed of the 3 subunits, that is the nitrogen base, a 5 carbon sugar, that may be ribose or the deoxyribose, and at least a phosphate, one phosphate group. If it is one, that is for example a adenine molecule, it is joined with a 1 phosphate group, so it forms a component AMP. If the 2 phosphate group, they are joined to adenine molecule, then they form a ADP, that is adenosine diphosphate, adenosine monophosphate. And if the 3 phosphate molecules, they are joined together to a adenine group, it is called as the ATP, that is the adenosine triphosphate, which is the powerhouse of the cell. ATP, they are the powerhouse of the cell. So as compared to this, the other nitrogen bases also, they compromises and they join together with phosphodiester bond and make a compound, that may be a GMP, that may be a GDP, or that may be a GTP. So you can see in the figure, at the 9th position of the purine of adenine, a bond is formed at the first position of a sugar, and at the 5th position of the sugar molecule, a phosphate group is attached by a phosphodiester bond. So this is a nucleotide which compromises by the 3 components, including a nitrogen base, a 5 carbon sugar, and at least 1 phosphorus molecule. Now we are going to see the DNA and RNA nucleotides. You can see only difference is that in DNA the deoxyribose sugar is present, and in RNA the ribose sugar is present. And the second main difference is in DNA nucleotides, the thymine nucleotide nitrogen base is present, and in RNA a uracil nitrogen base is present. The main difference between these two nucleotides are shown. Again, a base plus sugar, they compromises a nucleoside. Example, adenine plus ribose, adenosine. Adenine plus deoxyribose, that is deoxyadenosine. And if you are talking about nucleotide, it is sugar plus base plus phosphate group, which may be a DAMP, DDP, and DATP, that is the deoxyadenosine triphosphate, deoxyadenosine diphosphate, and the deoxyadenosine monophosphate. Now moving further, what the structure of DNA, which we are going to see in our next lecture. Before that, who invented this? Who told us the structure of DNA? Who told us that these are the components of the DNA? The DNA is double helical structure, it is anti-parallel strand, they are present. The nitrogen bases, they are bonded with the hydrogen bond. The backbone of the DNA, it is the sugar molecule and the phosphate molecule joined together by the phosphodiester bond. All this were told by the Watson and Crick. Before that, the various scientists told us that we are going to see a few key elements led to the discovery of the structure of DNA. DNA as the acidic substance present in the nucleus was first identified by Friedrich Mischer in 18th century, in 1868, and he named it as the nuclein. You can see his photo in the PPT or on the screen, that He was the scientist, he was the first scientist in the 18th century who told us that it is a nucleon. Next in 1953, James Watson and Francis Crick, they described very simple but famous double helical model of the structure of the DNA, which we study today was given to us by the Watson and Crick. Rosalind Franklin, you can see in the you can see on the screen, that is the Rosalind Franklin and the Morris Wilkins. They were also the scientists. Rosalind Franklin, she was the lady who first, before Watson and Crick, she first gave the X-ray crystallographic image of the structure of the DNA. She made marked advantages in X-ray diffraction techniques with the DNA. The diffraction pattern she obtained suggested several structure features of DNA. She told us the helical, more than one strand are present, 10 base pairs per complete turn they are present. She was the scientist who told all about this. Rosalind Franklin, she worked in the same laboratory as Morris Wilkins. She studied X-ray diffraction to study wet fibers of the DNA. You can see on the screen that X-ray crystallography given by the Rosalind Franklin photo, which gives us the structure of DNA, which is a double-stranded DNA. Then the Francis Crick and James Watson, you can see their pictures who gave us the structure of DNA. In 1953, J.D. Watson, an American biologist, and F.H.C. Crick, a British physicist, proposed the three-dimensional model of the physiological DNA. For this, Watson, Crick and Wilkins got a Nobel Prize in Medicine in 1962. Term DNA was given by Zacharias. James Watson and Francis Crick in 1953, they described the 3D structure. It consists of two polynucleotide chains of DNA wound around the same axis to form a right-handed double helix molecule, which we call as the B form of DNA, which is the right-handed DNA molecule. The various forms we are going to see in our next lecture. In that form, only Z form of DNA, it is the left-handed DNA molecule, otherwise all are the right-handed DNA molecules. Only some slight difference are present between the forms of DNA, which we are going to see in our upcoming lectures. So what we have seen till now, it is the types of nucleic acid, that is DNA and RNA, structure, that is the structure of nitrogen bases, nucleoside, nucleotide and the polynucleotide chain. In our upcoming lectures, we are going to see the main structure of DNA. We are going to see a polynucleotide chain, how it is formed in 5' to 3' and 3' to 5' direction. We are going to see which bonds they are actually involved in the structure of DNA. We are going to see the different forms of DNA. So these are the various precaps which we are going to see in our next lecture. So all of you, thank you for attending my lecture. If you have any difficulty, you can ask me. Thank you.